Hello, and welcome back to What is a Biology? I'm your host as always, Mr. Johnny Hopkins, and today we'll be covering more transcription factors. These guys are actually really interesting, so don't give me any flack for that. They're pretty cool. So first of all, we're just going to go through a general process for some generic transcription factors. Then we're going to go into something I kind of hinted at previously, something you know about the transcription factors messing around with the actual organization of the DNA, the chromatin. It's pretty interesting, as I have already said many times. And then we're going to go fishing. So, first of all, before we go fishing, let's remind ourselves what a transcription factor is. It's a factor that affects transcription. Oh yeah. So it's a protein that binds to DNA and then essentially stimulates the transcription of a certain gene, or it can also, repress the transcription of a certain gene. Today we'll be mostly talking about repressors, but first we're going to talk about activators. So one thing that usually happens is the transcription pre-initiation complex, which essentially it's this complex that associates with RNAP2 initiation factors, and it helps unwind the DNA. So first of all, remember, just IF means initiation factor. What is RNAP2? RNAP2 is the RNA polymerase that makes that makes mRNA, which makes proteins. 1 and 3 do not do that naturally. They work on just making RNA for the sake of RNA's sake. Then we've got the downstream promoter element, which is the DPE, which also facilitates the transcription for a tataless gene. So those genes are helped by this DPE, and the tata is just this little region that helps promote the transcription of a certain gene that a lot of transcription factors bind to. So there are a lot of different transcription factors which are initiation factors. You got TF2A, you got TF2B, you got TF2D. And TF2D notably is a 38 kilodalton protein that is also the tata binding protein, also known as a TBP. Now, TBP. So what usually happens is you have a TBP, the tata binding protein, bind to the tata box, and then goes to transcription factor 2B, then transcription factor 2F, and with RNA polymerase, then transcription factor 2H, which is a helicase, and then you have transcription occur. So that's the formation of this transcription pre-initiation complex. One thing to also know is that transcription factor 2A is necessary in vivo as well for this to occur, but the pipeline and everything was not quite well known at the time of the writing of the textbook material that I am using of Lotus et al. 6th edition, which is like 20 years old at this point. So now we're going to go on to essentially just the nature of repression. And so one thing that is notable is that transcription factors don't just necessarily bind to DNA, they can also affect the structure. So they can also interact with the histones, the nucleosomes, and if we remember correctly, a nucleosome is just a histone with DNA wrapped around it. So it's able to mess around with these nucleosomes to activate or repress them. So just one real quick remembrance is heterochromatin is very tightly packed, and so what that means is that you'll have an inactive gene if it's a heterochromatin, whereas if you have an active gene, it'll be euchromatin, which is just more like beads on a string, where you have essentially just a bead of a nucleosome stretched by some DNA, then a nucleosome, DNA, nucleosome, so on and so forth. And so one thing that is notable in activation in heterochromatin is that usually there's a methylation of the histone 3 subunits, and as well as some deacetylation in most of the lysines around. So one thing that one transcription factor that can go on is heterochromatin protein 1, HP1, which promotes the condensation naturally because it causes heterochromatin to occur. And so what happens is it binds the nucleosome and it adds a methyl into the lysine 9, removing the charge of lysine, which allows for tight action on the histone 3 subunit. So now we're going to go, so that's a general idea for how you can repress something. Now we're going to go to something a little more specific called Polycomb proteins. These guys will come up a lot later on, but this is more of a cellular level process that we'll talk about later. But Polycomb proteins help create chromatin structure, which is kind of what we're talking about, and they help cause repression. And what they usually do is they regulate Hox genes, and these Hox genes are going to come up in a while. And essentially, what Hox genes do is they encode regulatory transcription factors. What does that mean? They direct the development of different tissues in the embryo. They essentially help designate the differentiation of cells in our bodies to become, this one be a foot, this one be a stomach, this one be a brain. So as you can tell, I only got 2 of those brain cells because the Hox genes kind of messed up there. But so essentially these Hox genes develop and help make every single tissue in our body. And so they're regulated by Polycom proteins. And the pathway for this is you have a repressor, and that PRC, Polycom Protein 2, will bind to, and then you get some good old nucleosomes right down there. So what happens is that PRC2 binds to the nucleosomes, takes the K27, I believe it's a histone 2, B or A, does one of the histone subunits, not really that important for my purposes, and it takes that lysine and methylates it, so it reduces the charge, and then you have Polycom Protein 1, PRC1, come in and bind to all of those histones and cause condensation of the domains and makes them, not possible. So what it particularly does is it binds the methyls that we just add on about PRC2. So another way to do this is by deacetylating histone tails. Is that typically when active genes have highly acetylated tails of these histones, whereas when you remove these, deacetylation helps promote condensation, and usually the repressor can also bind the top box. And what that means is that, as well as the promoter proximal region, and what happens is that because it binds the top box and the promoter proximal region, the activator can no longer bind to the gene itself, which causes repression of the gene and condensation. So one thing that is one simple example is yeast GCN4 and GCN5. These guys actually help hyperacetylate the histone and terminal tails, which actually decondenses the chromatin, and so it opens up the chromatin and then facilitates TF2D to bind and cause actual transcription of a certain gene. So it can go and repress a gene by causing condensation of the histones and nucleosomes, or you can, and then when that happens, you can just take another transcription factor and just kind of say, hey, you shouldn't have done that, bro. Or it could go in and out of different sets of stages. So moving on, there's also another way that we can remodel the system of the chromatin as well. And what happens is you can have a chromatin remodeling complex. And the most typical ones that you're going to hear about is SWI/SNF. And these guys are actually homolog to the helicase protein. And what happens essentially is you have SWI, SNF bind to a certain part of DNA, and there's a nucleosome nearby, and essentially just translocates that DNA down. So essentially moves it all the way down that histone. And that's just probably an over-exaggeration. I have no idea how many base pairs it moves, but it probably move that much, maybe, I don't know. But just know that it translocates that so it can actually access different genes for different regions. One notable one is this actually occurs in the yeast HO gene, which encodes a nuclease. So essentially what happens is SWI5 binds to the enhancer, which then binds the SWI-SNF complex, as well as SAGA, which is a hyperacetylating complex of proteins that you can hear about a lot if you really, really want to get into it. And so what happens is that the chromatin is decondensed slightly by the SWI-SNF, and it's hyperacetylated by the SAGA unit. And then SPF will bind to the promoter proximal region, the PPR, and then RNA2P2 will assemble, and we'll have transcription of the HO gene. So now we're going to go fishing. Let me tell you, I'm excited just to say we're going fishing. It's cool. It sounds more fun than it actually is. So but it actually is fun. So what happens is that yeast can express, we can abuse this technique for different experiments, is yeast can express a DNA binding protein followed by a flexible linker with an activation domain. We're going to call this the bait domain. And then, without the activation domain, then you can have the cDNA library cloned into yeast with a strong activation domain and a flexible linker, and that's going to be the fish domain. So essentially, you have a DNA binding protein attached to a bait domain, and then you have a fish domain connected to an activation domain. So what essentially happens is that in the cell, the DNA binding protein will bind to a certain DNA, and then you have a bait domain, and then the fish domain, and the activation domain. So essentially when the bait and the fish match, you'll have them come together, and then the activation domain will find the DNA and activate the gene. So this is typically done with the HIS, the histidine synthase gene. So essentially when the activation domain happens, you get a pre-initiation complex to form. And so essentially when you have this matching bait and fish domain, you get colony formation of just a bunch of different yeast. So all this yeast comes together and you get beer. So you can use this to find different bait and fish pairs, as well as find other things about the genome in through these experiments of fishing. So now we're going to just do a real quick recap. You have a pre-initiation complex that has a bunch of different transcription factors, and it goes down a pipeline and you get transcription. Transcription factors can also interact with nucleosomes, and what happens is that you can deacetylate the histones or methylate them, and that will cause condensation of the domains. You have Polycom Proteins, which regulate Hox genes, which regulate the differentiation of the cells. And what happens with Polycom Proteins, essentially they methylate different lysines in these nucleosomes and cause repression by a large Polycomb protein complex to cause condensation and the repression of a lot of different genes that were used for differentiation of cells. And then you can also go back by hyperacetylating these genes and decondensing the chromatin. You have this guy called SWI-SNF that will remodel and rechange the positions of the DNA with respect to the nucleosomes. And you have this technique called fishing, where you have DNA binding protein, a bait domain, a fish domain, an activation domain. They can find matching bait and fish pairs, as well as find out other information about the structure of the genome. So going on from here, we will talk a bit more about transcription factors, as well as more about just other RNAPs, the 1 and the 3, the ones that aren't RNAP2. And then we'll go on to, after that little lecture, we'll probably go on and talk about some other stuff about genetic control that's not specifically, that's not gene level, but rather post-gene level, RNA level or protein level. So until next time, thank you.